Every autumn, a bird called the bar-tailed godwit takes off from the coast of Alaska and flies south. It does not stop. It does not eat. It does not sleep. For 11 straight days, this bird crosses 7,100 miles of open Pacific Ocean, burning through half its body weight in fuel, navigating with 0 landmarks beneath it, and landing exactly where it intended to land, in New Zealand. No GPS, no map, no training, just a 16-gram brain and a pair of eyes that might be running one of the most sophisticated quantum computers on Earth. That is not a metaphor. That is not poetic license. That is, according to a growing body of evidence from Oxford, Oldenburg, and a dozen other research institutions, literally what is happening inside the eyes of migratory birds. And the full story of how we figured this out is one of the strangest detective stories in the history of science. It involves a rejected physics paper, a husband and wife team who had to pretend they did not work together, a 3-year mystery solved by aluminum foil, a doctoral student who finally pulled a single protein out of a robin's eye and proved that quantum mechanics is not just something that happens in billion-dollar laboratories, and a military agency that funds stealth bombers quietly writing checks to people who study songbirds. It also involves one of the deepest questions in all of physics. Can quantum effects survive in the warm, wet, chaotic environment of a living cell? For most of the 20th century, the answer was an emphatic no. Quantum coherence, the delicate superposition of states that makes quantum computers work, was supposed to collapse instantly at biological temperatures. You need temperatures near absolute zero. You need near perfect vacuum. You need isolation from every stray vibration and photon. A bird's eyeball bouncing along at body temperature in a skull being rattled by flight muscles is about as far from those conditions as you can possibly get. But the birds did not read the textbook, and the evidence now suggests they have been running quantum technology in their eyes for tens of millions of years. Before any of that made sense, though, we had to spend centuries having absolutely no idea how birds knew where they were going. Think about this for a second. Humans have known that birds migrate since at least the time of Aristotle. The ancient Greeks watched swallows disappear every fall and genuinely believed they hibernated in the mud at the bottom of lakes. That was the leading scientific theory for roughly 2,000 years. In the 1700s, a naturalist named Charles Morton proposed that birds migrated to the moon. He did the math. He calculated the journey would take about 60 days. People took this seriously. In 1822, a white stork was found in Germany with an arrow from Central Africa embedded in its neck, still alive, providing the first physical evidence that European birds traveled to Africa. Before that, nobody was sure. The point is not to laugh at the ancients. The point is that bird migration is so improbable, so difficult to explain, that otherwise intelligent people preferred lunar travel and mud hibernation to admitting they had no idea what was going on. And the scale of the problem only got worse the more we learned. During World War II, the British military parachuted over 16,000 homing pigeons into occupied Europe, strapped to agents and resistance fighters, and those birds flew home across hundreds of miles of unfamiliar territory with a success rate that embarrassed the Royal Air Force's actual navigation equipment. Arctic terns migrate from pole to pole and back again every year, covering roughly 44,000 miles. Black pole warblers, tiny songbirds weighing barely half an ounce, fly non-stop over the open Atlantic Ocean from northeastern North America to South America, 88 hours straight over featureless water. And they do not take the same route twice. Whatever compass these animals carry it is not a simple one. The scientific story of cracking that compass begins in the early 1950s at the Max Planck Institute in Wilhelmshaven, Germany, where a researcher named Gustav Kramer figured out the first piece of the puzzle. He put European starlings in circular cages with a view of the sky and the birds oriented in their preferred migratory direction. Then he used mirrors to shift the apparent position of the sun, and the birds shifted their orientation to match. They were using the sun as a compass. This was a genuine breakthrough, but it immediately raised a harder question. What about birds that migrate at night? That question got answered about a decade later by Stephen Emlen at Cornell. Emlen invented a beautifully simple device called the Emlen funnel, basically a cone-shaped cage with ink pads on the bottom and paper on the sloped walls. When a restless migratory bird hops in its preferred direction during the migratory season, a behavior called sugunru that is hardwired into their biology, it leaves ink marks that tell you exactly which way it wants to go. The device costs almost nothing to build and has been used in thousands of experiments since. It is one of those elegant inventions that makes you wonder why nobody thought of it sooner. Emlen took indigo buntings into a planetarium and showed them an artificial night sky, the birds oriented by the stars. Then he did something clever. He raised a batch of young buntings under an artificial sky that rotated around Betelgeuse instead of Polaris. Those birds grew up treating Betelgeuse as north. Star navigation, it turned out, was not hardwired. It was learned. Young birds watched the sky during their first summer, figured out which point the stars rotated around, and used that as their reference for the rest of their lives. This was published in a series of papers between 1967 and 1970, and it remains one of the most elegant experiments in animal behavior. So by the early 1960s, we knew about two compasses: the sun compass for daytime, the star compass for nighttime. Both were impressive, and both had obvious limitations. Overcast skies would knock out both of them. Yet birds navigated perfectly well through clouds, through rain, through conditions where neither the sun nor a single star was visible. Something else was going on, something we could not see, could not measure, and for a long time could not even imagine. On October 12, 1963, in a basement laboratory at Goethe University Frankfurt, a graduate student named Wolfgang Wiltschko did something that would eventually upend our understanding of animal senses. Working in the laboratory of Friedrich Merkel, Wiltschko placed European robins inside a steel chamber that partially shielded Earth's magnetic field. He was not specifically looking for a magnetic sense. He was studying migratory restlessness, the agitated hopping that captive birds display during migration season. But when he manipulated the magnetic field inside the chamber, the birds changed their preferred direction. They were using the residual magnetic field as a compass. Wiltschko tested it again and again. The result held. He shifted the field. The birds shifted with it. He rotated it 90 degrees. They rotated 90 degrees. This was the first experimental evidence that birds could sense magnetism, and it was met with, as Wiltschko later put it, the highest skepticism. Other researchers openly doubted the results. Some suggested the birds were responding to subtle temperature gradients or air currents, or something in the room that Wiltschko was not controlling properly. Nobody believed him. And honestly, why would they? Earth's magnetic field is absurdly weak. We are talking about roughly 50 microtesla, which is about 1 million times weaker than a refrigerator magnet. The energy of interaction between that field and any known biological molecule is roughly 1,000,000 times smaller than the thermal noise constantly rattling every atom in a living cell. Suggesting that a bird could detect this field was like suggesting you could hear someone whisper from across a football stadium during a jet engine test. The physics just did not seem to work, but Wiltschko kept at it. In 1968, he met a first-year student named Roswitha. They married that year and began one of the most productive partnerships in the history of biology, though for years they had to publish separately because, as Wolfgang later explained, we had to separate, at least for the public. Academic politics in the 1960s was not kind to married couples working together. Roswitha Wiltschko would go on to become a professor in her own right and publish over 300 papers on magnetoreception. But the early years required navigating a system that did not quite know what to do with two people sharing a last name on the same paper. Their landmark 1972 paper in Science, Magnetic Compass of European Robins, delivered a finding that nobody expected. The bird compass was not a polarity compass. It did not detect which direction was magnetic north and which was south, the way a handheld compass does. Instead, it was an inclination compass. The birds were detecting the angle at which Earth's magnetic field lines dip into the ground. This matters enormously. And here is why. At the magnetic equator, field lines run parallel to Earth's surface. As you move toward the poles, the lines tilt more steeply downward. In Frankfurt, the inclination angle is about 66 degrees. At the North Pole, it is 90 degrees, straight down. By detecting this tilt angle relative to gravity, birds can distinguish poleward from equatorward without ever needing to know where north is. Flip the polarity of the magnetic field in the lab, and the birds do not get confused. Flip the inclination, and they immediately reverse direction. This was wild, but acceptance was painfully slow. The real turning point came in 1976, when the Wiltschkos collaborated with Emlen at Cornell on a double-blind study using indigo buntings that was published in Science. Wolfgang recalled the shift simply. People then believed his earlier work. Sometimes it takes a big name at an American university to make the world pay attention to a finding from Frankfurt. Through the 1980s and 1990s, the Wiltschkos systematically mapped the properties of the magnetic compass, and each new finding was stranger than the last. The compass only works under blue and green light, up to about 565 nanometers wavelength. Under red light, it fails completely. This was a major clue pointing toward a photochemical mechanism, something that needed specific wavelengths of light to function. By 2002, they demonstrated the compass is lateralized, functioning only through the right eye. Cover the right eye, and the magnetic compass disappears. Cover the left eye, and it works fine. Whatever was detecting the magnetic field was not just in the eyes. It was specifically in the right eye, processed by the left hemisphere of the brain. All of these findings were clues: the light dependence, the inclination sensitivity, the lateralization. They painted a picture of a sensor that was photochemical, located in the eye, and wired into the visual processing system. But what kind of biological molecule could detect a force so weak that no known physical mechanism could explain it? The answer, when it finally came, arrived from the last place anyone was looking: quantum physics. And it came from a man whose idea was so far ahead of its time that the most prestigious journal in the world told him to throw it in the trash. Klaus Schulten was born in 1947 in the Ruhr Valley region of Germany and earned his PhD at Harvard under Martin Karplus, who would later win the Nobel Prize in Chemistry in 2013. Schulten was a theorist. the kind of physicist who worked with equations rather than equipment. And he had a gift for seeing connections between fields that other people kept in separate boxes. In 1974, working at the Max Planck Institute in Göttingen under a physical chemist named Albert Weller, Schulten made a discovery about how magnetic fields could influence chemical reactions through quantum spin dynamics. The specific problem involved something called fast triplets, anomalously rapid chemical reactions that nobody could explain. Schulten realized that magnetic fields could modulate the quantum spin states of electron pairs, altering which chemical pathway the reaction followed. The backstory is almost comically dramatic. His lab manager, Hubert Sterk, insisted the theoretical prediction was wrong. Schulten pushed back. Sterk eventually admitted, with apparent reluctance, that he had already done the experiment and found a small effect exactly matching what Schulten had predicted. A disaster turned into a happy moment, Schulten later recalled, because I knew what to look for. He did not. Schulten immediately saw the implications for biology. If magnetic fields could alter the outcome of chemical reactions by influencing the quantum spin states of electrons, then maybe that was how birds did it. Maybe there was a chemical reaction happening inside the bird where the products depended on which way the magnetic field pointed. He wrote a paper proposing exactly this and submitted it to Science. The rejection letter contained a line that Schulten could quote from memory 30 years later. A less bold scientist would have designed this piece of work for the wastepaper basket. He published it anyway in 1978 in an obscure German journal called Zeitschrift für Physikalische Chemie, with co-authors Charles Swenberg and Albert Weller. The paper proposed what is now called the radical pair mechanism, the idea that a chemical reaction involving two molecules with unpaired electrons could act as a biological compass because Earth's magnetic field influences the quantum spin states of those electrons. The paper was largely ignored for more than 20 years. Most physicists did not read obscure German chemistry journals. Most biologists did not understand quantum spin dynamics, and the whole idea sounded like science fiction. Schulten moved to the University of Illinois at Urbana-Champaign in 1988, where he founded the Theoretical and Computational Biophysics Group at the Beckman Institute. He built one of the world's great computational biology laboratories, eventually leading a team of over 30 researchers and developing the VMD and NAMD software packages used by more than 300,000 scientists worldwide for molecular dynamics simulations. His wife, Zaida Luthi Schulten, became a chemistry professor at Illinois. They too had been forced to navigate the academic two-body problem, the same institutional awkwardness that had plagued the Wiltschkos. It is a recurring theme in this story. The science of how birds navigate as pairs was, ironically, advanced by human pairs who were told they could not work together. Through all of it, Schulten kept working on his magnetic bird idea on the side, publishing occasionally, refining the theory, waiting for the world to catch up. If you want to solve a problem, he once said, it takes a lot. That might be the understatement of the century. The world caught up in the year 2000, and the person who bridged the gap was Schulten's own graduate student, Thorsten Ritz. Their paper in the Biophysical Journal, titled A Model for Photoreceptor-Based Magnetoreception in Birds, did two things that transformed the field. First, it made the radical pair proposal accessible to biologists for the first time, translating the dense quantum physics into language that experimentalists could use. Second, and more importantly, it named a specific molecule, Cryptochrome, a blue light photoreceptor found in the retinas of birds. Cryptochrome had been discovered in the late 1990s as a protein involved in circadian rhythms, the internal clock that tells organisms when to sleep and wake. Plants have it, fruit flies have it, humans have it. But the version found in bird retinas, particularly the variant called Cryptochrome 4, had properties that made it uniquely interesting. It absorbs blue light. It contains a molecular component called flavin adenine dinucleotide that can generate radical pairs when activated by light. And it is physically located in the photoreceptor cells of the retina, exactly where a light-dependent magnetic sensor would need to be. The logic was elegant. Whatever molecules served as the magnetic sensor had to satisfy several requirements. It had to be activated by light, since the Wiltschko had shown that the magnetic compass only works under blue and green light and fails completely under red. It had to be located in the eye, since the compass signal appeared to travel through the visual system. It had to be capable of forming radical pairs, meaning pairs of molecules with unpaired electrons whose quantum spin states could be influenced by a magnetic field. And it had to be expressed in migratory species. Cryptochrome checked every box. Peter Hoare at Oxford, who would later become one of the field's most important theorists, along with Moritzen, would describe this paper as having transformed an interesting curiosity into an intriguing possibility. Research in the field skyrocketed almost overnight, but this was still just a hypothesis, a very good hypothesis, and the scientific community had been burned before by elegant ideas that turned out to be wrong. What Schulten's detractors wanted was evidence, experimental evidence. And Ritz, now at UC Irvine, to his enormous credit, designed the experiment that could provide it. The logic went like this. If the bird compass works through radical pair chemistry, then the quantum spin states involved would oscillate at specific frequencies determined by the local magnetic environment. Those frequencies happen to fall in the megahertz range, matching the Larmor precession frequency of electrons in Earth's magnetic field, roughly 1.4 megahertz. That means if you bombarded a bird with a weak oscillating magnetic field at that exact frequency, you should be able to disrupt the compass, not by overpowering it with a stronger field, but by scrambling the quantum coherence that makes it work. It would be like playing a musical note at exactly the right pitch to shatter a glass, except the glass in this case was a quantum state inside a protein. Ritz had to convince the Wiltschko to run the experiment, which was not easy. They had spent their careers demonstrating that the magnetic compass existed, and now this young physicist was proposing an experiment that might break it. Then he had to wait 7 months for the migratory season, because migratory birds only show compass orientation when they are in the mood to migrate. You cannot rush a robin. The results, published in Nature in 2004, were exactly what the theory predicted. Weak radio frequency fields at the Larmor frequency disrupted robin orientation completely, while the same fields applied parallel to the static field had no effect. The specificity was remarkable. It was not just any electromagnetic interference. It was interference at exactly the frequency that radical pair theory predicted would matter. A broadband field that included the Larmor frequency disrupted the compass. A narrowband field at the wrong frequency did not. This was the first direct experimental evidence that the bird compass might genuinely operate through quantum mechanics, and it gave the field something it had never had before, a testable, falsifiable, mechanism-specific prediction that had been confirmed. But the most extraordinary part of this whole story is what is actually happening at the molecular level. Because if you really dig into the mechanism, it is one of the most improbable things in all of biology. And I want to walk through it carefully, because the details matter. Here is how it works. A photon of blue light, wavelength around 450 nanometers, enters the bird's eye and hits a cryptochrome 4 protein sitting in the retina. Inside that protein is a molecule called flavin adenine dinucleotide, or FAD, curled up in a U-shaped pocket. Think of it as a molecular antenna waiting for the right signal. The FAD absorbs the photon and gets excited, jumping to a higher energy state. Within about half a picosecond, which is half a trillionth of a second, the excited FAD yanks an electron away from a neighboring amino acid called tryptophan. Now you have two molecules sitting very close together, each with an unpaired electron. This is the radical pair. Two molecules, each missing an electron, each in a chemically reactive state. This part is just chemistry. Unusual chemistry, but chemistry. And here is where quantum mechanics enters. And honestly, this is where the story goes from interesting to genuinely mind-bending. Those two unpaired electrons are born in what physicists call a singlet state. Their spins are anti-parallel, pointing in opposite directions, perfectly correlated. They are, for all practical purposes, quantum entangled. Now, in normal chemistry, you would not care about this. The spin state of electrons matters in a few niche areas of physical chemistry, but it is not the kind of thing that keeps biologists up at night. Except in this case, the spin state is everything, because this electron pair does not just sit in the singlet state. It exists in a quantum superposition, simultaneously in the singlet state and in something called the triplet state, oscillating between the two at frequencies in the megahertz range, driven by interactions with nearby hydrogen and nitrogen nuclei called hyperfine couplings. The system is coherent. It is maintaining quantum superposition at body temperature in a wet, noisy biological cell, and that superposition is oscillating back and forth millions of times per second. Let me say that again because it is important. A protein inside a bird's eye is maintaining quantum coherence at 37 degrees Celsius. Quantum computers need to be cooled to 15 millikelvin to do this. The bird does it at body temperature, in flight. But the electron does not just sit there, it hops. The cryptochrome protein contains a chain of 4 tryptophan amino acids, one after another. Like stepping stones, the electron hole migrates down this chain, jumping from the first tryptophan to the second, to the third, to the fourth, each hop increasing the distance between the two unpaired electrons. The first tryptophan sits about 7.4 angstroms from the FAD. The third sits about 18.2 angstroms away. And this increasing distance is critical because it weakens the coupling between the electron spins and allows Earth's magnetic field, weak as it is, to influence the quantum dance. What the magnetic field does is subtle but decisive. Through something called the Zeeman effect, the field lifts the degeneracy of the triplet substates, making the rate of singlet to triplet conversion depend on the angle between the magnetic field and the molecular axis of the cryptochrome. Cryptochrome molecules oriented one way relative to the field will have a different singlet to triplet ratio than molecules oriented another way. And here is the key. Only singlet state radical pairs can recombine back to the ground state. Triplet recombination is forbidden by the rules of quantum mechanics. So, the chemical product of this reaction, the thing the bird's nervous system actually detects, depends on which way each cryptochrome molecule is pointing relative to Earth's magnetic field. Now remember that these cryptochrome proteins are distributed across the bird's retina, which is roughly spherical. Different proteins face different directions. Some face toward the magnetic field lines, Some face away, some face sideways. Each one produces a slightly different chemical signal, depending on its orientation. The result is a pattern, a kind of visual overlay, probably something like a bullseye or a band of light and shadow, superimposed on the bird's normal vision. When the bird turns its head, the pattern shifts. The bird literally sees the magnetic field. Try to imagine what that would be like. You wake up one morning and you can see gravity. Not feel it, see it. Every time you turn your head, a translucent pattern of light and dark shifts across your visual field, telling you exactly which way the mass of the Earth is pulling. Now replace gravity with magnetism, and you have something close to what a European robin may experience. The pattern would change as the bird flies north or south because the inclination angle changes with latitude. It would look different at the equator than at the poles, and it would be present all the time as long as light was hitting the eye, a permanent navigational display built into the bird's own vision. This has a profound implication. The bird does not need to consciously process magnetic information as a separate sense. It might just be part of what seeing looks like. The magnetic field would be woven into the visual world, the way color is woven in, or brightness. You do not consciously think about the fact that leaves are green. You just see them. A robin might not consciously think about the magnetic field. It might just look outside and know which way is south. If this sounds insane, I want you to understand something. This is not fringe science. This is not speculation. This is the leading hypothesis for how migratory birds navigate, supported by decades of behavioral experiments, a landmark paper in Nature, and tens of millions of euros in government funding. And the evidence kept getting stronger. In 2005, a researcher named Henrik Moritzen at the University of Oldenburg discovered something that nobody was expecting. Moritzen is the kind of scientist who does not just study birds in the lab. He has banded more than 6,000 of them personally. He grew up in Denmark as a bird watcher, spent years studying their behavior in the field, and eventually became a neuroscientist because he wanted to understand what was happening inside their heads. His contribution to this story is immense, and it begins with an accidental discovery. Using a gene marker called Zenk that lights up in recently active neurons, Moritzen and Duke University neuroscientist Eric Jarvis found a tight cluster of neurons in the brains of migratory European robins that activated specifically during nighttime magnetic orientation. They called it Cluster N. It sits in the visual wulst, a part of the bird brain that processes visual information, which was the first hint that whatever magnetic signal these birds were receiving was being processed as though it was something they could see. When they tested non-migratory species like zebra finches under identical conditions, Cluster N showed no activity at all. The neurons only fired in migratory birds, and only when those birds were actively orienting at night. A 2007 study by Hayes and colleagues traced the neural pathway and found a direct connection from cryptochrome expressing neurons in the retina through the visual thalamus all the way to Cluster N. There was a physical wire running from the eye to the brain, and it carried the magnetic signal. But the definitive proof came 2 years later. 4 years after the initial discovery, Moritzen's team published a paper in Nature that delivered perhaps the strongest evidence yet for the eye-based quantum compass. They surgically destroyed Cluster N in both hemispheres of a group of robins. Those birds completely lost their magnetic compass. They could not orient magnetically at all, but they could still navigate by the sun. They could still navigate by the stars. Only the magnetic compass was gone. Then they took a different group of robins and severed the trigeminal nerve, which carries signals from the beak to the brain, cutting the connection to any potential iron-based magnetite sensors in the upper beak. Those birds navigated magnetically without the slightest difficulty. The compass signal was coming through the eyes, traveling through the visual system to cluster N, exactly as the radical pair hypothesis predicted. The beak had nothing to do with it. Moritzen now says, "I am 99% sure that cluster N is a magnetic compass center in the bird's brain. The 1% of doubt is just good scientific practice." But there is another experiment from Moritzen's lab that might be the most dramatic of all, and it started as a total disaster. Between 2004 and 2007, Moritzen's team spent 3 consecutive migratory seasons trying to test European robins in wooden huts on the University of Oldenburg campus. And for 3 years straight, the birds could not orient. They were completely lost. The team changed the food. They changed the cages. They adjusted the light cycles. They tweaked the magnetic field parameters. Nothing worked. 3 years of failed experiments, which in academia is the kind of thing that ends careers. Then, a graduate student named Nils Lass Schneider suggested something that sounded almost absurdly simple. What if they covered the huts with grounded aluminum sheeting? Basically, what if they built a Faraday cage? The idea was that maybe electromagnetic noise from the campus was interfering with the bird's compass. They tried it. The birds immediately recovered their magnetic orientation perfectly, as if the 3 years of failure had never happened. When they disconnected the grounding wire from the aluminum sheeting, the birds lost orientation again. Reconnected it, orientation came back. They tested it over and over, on and off, on and off. The result was binary and instant. Shielded from electromagnetic noise, the birds could navigate. Exposed to it, they were blind. They moved the birds to an unshielded shelter several kilometers outside the city, away from the campus electronics and radio transmitters, and the birds oriented correctly without any shielding at all. The city was the problem, or more precisely, the invisible soup of electromagnetic radiation that blankets every modern city was the problem. This was published as a cover story in Nature in May 2014, and the implications were staggering. Anthropogenic electromagnetic noise, the kind of radio frequency pollution generated by every consumer electronic device on the planet, in the frequency range of 2 kHz to 5 MHz, was disrupting the quantum compass at intensities 1,000th of the levels the World Health Organization considers safe for humans. The magnetic component of the noise was just 15 nanotesla. That is unbelievably faint, and it was enough to blind these birds to Earth's magnetic field. A follow-up study in 2023 pinned down the cutoff frequency at roughly 116 MHz. Everything below that, including AM radio frequencies, interferes with the compass. Mobile phone frequencies above that threshold do not. This is a very specific prediction that matches what you would expect from a radical pair mechanism with a particular set of quantum properties. And it is worth noting that this research was partly funded by DARPA, the United States military's advanced research agency, through a program called QEF-BE, which stands for Quantum Effects in Biological Environments. The American military is interested in quantum bird compasses. Think about that for a second. But all of this was still circumstantial. Behavioral experiments showing birds can be disoriented, brain regions that light up during magnetic navigation, radio frequency interference at predicted frequencies. What nobody had done was take the actual molecule out of a bird's eye and show that it responds to magnetic fields in a test tube. Until 2021. Before I get to that paper though, I need to tell you about Peter Hoare, because his role in this story is just as important as Morrison's. Hoare is an Oxford chemist who has spent virtually his entire career at the university, starting as an undergraduate in 1973 and joining the faculty in 1983. His specialty is spin chemistry, the study of how the quantum spin states of electrons influence chemical reactions. For most of his career, this was a niche field. Then, the Bird compass came along, and suddenly spin chemistry was the key to one of the biggest mysteries in biology. Hoare brought theoretical rigor to the field. He co-authored the definitive review paper with Morrison in 2016 in the Annual Review of Biophysics, laying out the state of the evidence and the remaining questions. His group created the first artificial chemical compass, a man-made molecular system sensitive enough to respond to Earth's magnetic field, and he made a statement that captures the stakes of this entire scientific enterprise. If we can prove that Cryptochrome 4 is the magnetic sensor, he said, we will have demonstrated a fundamentally quantum mechanism that makes animals sensitive to environmental stimuli 1,000,000 times weaker than previously thought possible. Together, Hoare and Morrison hold an €8.6 million European Research Council synergy grant called Quantum Birds. The Oxford Oldenburg axis, as some in the field call it, has become the intellectual engine of the entire discipline. Morrison brings the biology, Hoare brings the physics, and between them, they finally made it possible to test the hypothesis at the molecular level. The paper that changed everything was published in Nature in June of 2021. The lead author was Jingjing Xu, a doctoral student in Morrison's group. What Xu did was extract the genetic code for Cryptochrome 4a from European robins, produce the protein in bacteria in large quantities, purify it, and ship it to Oxford, where the groups of Christiane Timmel and Stuart Mackenzie used magnetic resonance and optical spectroscopy to test whether it was magnetically sensitive. It was. 3 findings emerged from that paper, and each one was significant. First, purified robin Cryptochrome 4 showed magnetically sensitive photochemistry in vitro. The molecule responded to magnetic fields. Second, the Cryptochrome 4 from migratory European robins was more magnetically sensitive than Cryptochrome 4 from non-migratory chickens and pigeons. The migratory species had the better compass. Third, when the researchers mutated each of the 4 tryptophan residues in the chain, swapping them for a different amino acid that cannot support the radical pair mechanism, magnetic sensitivity decreased systematically. The tryptophan chain that the theory predicted was essential turned out to be essential. This was the moment the field had been waiting for since 1978, 43 years after Klaus Schulten submitted his paper to Science and was told to throw it in the trash. His core idea was confirmed in the most prestigious scientific journal in the world. The protein he predicted should exist, in the location he predicted it should be, doing the chemistry he predicted it should do, was real. He did not live to see it. Schulten died on October 31st, 2016, at the age of 69, 5 years before the paper was published. His wife San continued at Illinois. His students, including Ritz, continued building on his work. The VMD and NAND software he developed are still used by hundreds of thousands of researchers. But the man who first imagined that a bird could sense a magnetic field through quantum mechanics never got to read the paper that proved him right. The research community responded with a mix of excitement and cautious reservation. 6 putative protein interaction partners of Cryptochrome 4 in the robin retina were identified, including cone-specific G proteins and long-wavelength sensitive opsin, suggesting that the magnetic sensor might couple directly to the same signaling cascade that processes normal vision. The bird might not just detect the magnetic field through its eyes. It might see it through the same neural machinery it uses to see light and color. But the story does not end there, because even with the 2021 result, significant problems remained. The experiments used magnetic fields about 40 times stronger than Earth's field. Nobody had shown the mechanism working in a living bird's eye. The actual pathway from radical pair chemistry to nerve signal remained completely unknown, and there were serious people making serious arguments that the whole thing might still be wrong. The most prominent alternative explanation comes from Joseph Kirschvink at Caltech, who has championed magnetite-based magnetoreception since the early 1980s. The idea is straightforward. Some animals contain tiny crystals of magnetite, an iron oxide mineral, essentially biological compass needles. These crystals could physically pull on ion channels, opening or closing them, depending on which way the magnetic field points. Iron-rich particles were indeed found in the upper beaks of pigeons, and for a while, this seemed like it might be the answer. Then, in 2012, David Keyes and colleagues at the Institute of Molecular Pathology in Vienna published a paper in Nature showing that those iron-rich cells in pigeon beaks were actually macrophages, immune cells, not neurons, not magnetosensors, just white blood cells that happen to accumulate iron because that is what macrophages do. This was embarrassing for the magnetite camp. Years of research had been based on the assumption that these cells were specialized magnetoreceptors, and it turned out they were the biological equivalent of a janitor carrying a bag of iron filings. The Wiltschkos have contested the finding, arguing that Keyes' team used methods unsuited for detecting superparamagnetic particles and behavioral evidence for magnetite-based sensing, particularly from magnetic pulse experiments that temporarily disorient adult migrants, remains robust. The most likely answer, and the one that most researchers now accept, is that both mechanisms exist. This is not a contradiction. The radical pair system in the eyes provides the compass, telling the bird which direction to fly. A magnetite or other system in the beak or elsewhere provides the map measuring field intensity to help determine position. Think of it this way. The compass tells you which way is south. The map tells you how far south you still need to go. You need both. In 2025, Keyes' group actually published evidence for a third mechanism entirely, involving electromagnetic induction in the pigeon inner ear, mediated by specialized type 2 hair cells expressing voltage-gated calcium channels. They called it a dark compass because it works without light. It turns out that bird navigation might use not just 2 compasses, but 3. These animals are carrying more navigation equipment than some naval vessels. There is also a legitimate scientific debate about whether the radical pair hypothesis has become what philosophers of science call a ruling hypothesis. This is a concept that goes back to the geologist Thomas Chamberlain in 1890, who warned that when scientists become too attached to a single theory, they unconsciously interpret all evidence in its favor. A provocative 2025 commentary in the Journal of Experimental Biology by Wortman, Fittak, and Nathan made exactly this argument about radical pair magnetoreception. They pointed out that no sensory receptor for magnetoreception has been unequivocally demonstrated in any organism. Not in birds, not in fish, not in insects. After decades of research, we still have not watched the compass work in a living animal. There remains a gap of several orders of magnitude between Earth's field strength and the sensitivity demonstrated in the test tube. And the field may be suffering from confirmation bias. This is a serious critique, and responsible scientists take it seriously. Science advances fastest when smart skeptics push back against popular ideas, and the skeptics here are not cranks. They are raising real, methodological, and philosophical objections. And then there is the Drosophila problem, which shook the field more than anyone expected. In 2008 and 2009, a team led by Stephen Reppert at the University of Massachusetts published papers in Nature showing that fruit flies could sense magnetic fields using Cryptochrome. These papers were widely cited as evidence that Cryptochrome-based Magnetoreception was widespread across the animal kingdom, not just limited to birds. If tiny fruit flies could do it, the mechanism was clearly ancient and robust. But in 2023, a team that included Moritzen and Hoare themselves published a devastating rebuttal. Also, in Nature, after testing over 97,000 individual fruit flies across 6 years of painstaking experiments, they found no evidence whatsoever for magnetically sensitive behavior in Drosophila. Not a hint, not a statistical trend, nothing. They suggested that many, if not all, of the original results were false positives, possibly arising from subtle environmental variables that the earlier experiments had not controlled. Reppert and others have pushed back hard. Charalambos Kyriakou at the University of Leicester published a rebuttal in Nature in 2024 that raised a particularly uncomfortable point. Earlier work had shown that Drosophila magnetoreception seemed to require only a tiny fragment of the cryptochrome protein, the last 52 amino acids, which completely lacks both the FAD binding domain and the tryptophan chain that are supposedly essential for the radical pair mechanism. If the mechanism works differently in flies than in birds, then maybe the negative results say nothing about the bird compass. But if the original fly results were just noise, then one major pillar of evidence for the universality of cryptochrome-based navigation has been quietly knocked out. Even Peter Hoare, who has spent decades building the theoretical framework for radical pair magnetoreception, has expressed caution that is worth quoting. It is an attractive idea that nature has adopted and optimized fundamentally quantum phenomena for its own purposes, he has said. But my sense is that it has really not been established yet that any of these phenomena are genuinely quantum mechanical. When the person who has done more than almost anyone to prove the hypothesis tells you the case is not yet closed, that means something. So where does that leave us? Is the quantum bird compass real, or is it an elaborate mirage? The answer, as of right now, is that the evidence has gotten dramatically stronger since 2021, and the last few years have produced a cascade of breakthroughs that have transformed this from a tantalizing hypothesis into something that looks increasingly like settled science. The first major development was theoretical. The biggest unsolved problem with the radical pair mechanism was sensitivity. Earth's magnetic field is absurdly weak, about 50 microtesla. The energy of its interaction with electron spins is roughly 1,000,000 times smaller than the thermal energy sloshing around in a biological cell. How can a signal that faint possibly be detected? In December 2024, Daniel Katnig's group at the University of Exeter published a paper in Nature Communications demonstrating that something called the quantum Zeno effect could solve this problem. The quantum Zeno effect is one of the stranger predictions of quantum mechanics. It says that if you observe a quantum system frequently enough, you can freeze it in its current state. Imagine you are watching a pot of water. In the quantum world, if you watch it closely enough, it literally never boils. What Katnig showed was that certain radical pair configurations, even tightly bound ones, where the electrons are very close together, separated by only about 4.5 angstroms, can be made sensitive to Earth-strength magnetic fields through this mechanism. Frequent chemical measurement of the radical pair state paradoxically preserves the quantum coherence that enables magnetic sensing. This overturned the long-standing assumption that only well-separated radical pairs, the kind generated by the tryptophan chain in Cryptochrome 4, could work as compass sensors. It opened the door to a much wider range of possible compass chemistries and removed one of the biggest theoretical obstacles to the hypothesis. Then, in September 2025, the same group showed that something called chirality-induced spin selectivity, or CISS, further enhances magnetic sensitivity. Here is the idea. Chiral molecules are molecules that have a preferred handedness, like a left shoe versus a right shoe. They are mirror images of each other, but cannot be superimposed. Proteins, including Cryptochrome, are inherently chiral because they are built from amino acids that all have the same handedness. What the Exeter team showed was that this handedness has a quantum consequence. Chiral molecules tend to transmit one electron spin orientation more readily than the other, and this introduces triplet character into the initial radical pair state, amplifying the magnetic signal. The protein's own molecular geometry, its inherent twist, was helping make the compass work. Evolution did not just stumble onto a quantum mechanism. It optimized the quantum mechanism using the protein's own shape. On the evolutionary front, a 2024 study in Proceedings of the Royal Society B analyzed Cryptochrome 4 genes across 363 bird species, the first large-scale evolutionary investigation of the compass molecule. This was the kind of study that nobody could have done even 10 years ago because it required genomic data from hundreds of species that simply was not available. The results were striking. Cryptochrome 4 turned out to be far more genetically variable than Cryptochrome 1 and Cryptochrome 2, which serve conserved circadian clock functions and barely change across species. This is exactly what you would expect if Cryptochrome 4 is under active selection pressure for environmental sensing rather than just running a clock. Different species face different magnetic environments, different migration distances, different navigational challenges, and their compass molecules are diversifying accordingly. Even more striking, the Tyranny suborder of passerines, a group that includes flycatchers, antbirds, and ovenbirds, appears to have lost the Cryptochrome 4 gene entirely. An entire lineage of birds just does not have it. This creates a natural experiment that researchers are now exploiting to test whether loss of Cryptochrome 4 correlates with changes in migratory behavior. In August 2025, Xu and colleagues confirmed in the Journal of the Royal Society Interface that the alternative isoform, Cryptochrome 4b, does not bind FAD, effectively ruling it out as a magnetoreceptor and consolidating Cryptochrome 4a as the sole radical pair candidate. The compass molecule now has a name and an address. But the breakthrough that may ultimately matter most for the world outside ornithology was published in Nature in January 2026. Just a few weeks ago, Gabriel Abrahams, Harrison Steele, Christian Timmel, and colleagues at Oxford demonstrated something called MAGLOV, short for magnetosensitive fluorescent proteins. Using directed evolution, essentially a kind of accelerated natural selection in the lab, they created proteins that exhibit optically detected magnetic resonance inside living bacterial cells at room temperature with single-cell detection sensitivity. These are engineered quantum sensors based on the same radical pair mechanism that birds use, and they work at body temperature in living cells without the cryogenic cooling that conventional quantum sensors require. The applications are extraordinary. The team demonstrated spatial localization using magnetic field gradients, which is essentially a biological version of MRI that works at the cellular level. They showed molecular microenvironment sensing, meaning these proteins can detect the chemical conditions inside individual cells. They showed multiplexed bioimaging. Harrison Steele put it this way: "What blows me away is the power of evolution. We do not yet know how to design a really good biological quantum sensor from scratch, but by carefully steering the evolutionary process in bacteria, nature found a way for us." The irony of that statement is almost too perfect. We spent decades telling ourselves that quantum effects could not survive in biology. Now we are using biology to build quantum devices that work at temperatures where our own quantum technology fails. This is the moment where quantum bird navigation stops being just a beautiful piece of biology and becomes a technology story. If the radical pair mechanism really works the way we think it does, then evolution has been running room temperature quantum sensors for tens of millions of years. Sensors that human engineers are only now learning to replicate. DARPA has multiple programs investigating biological quantum sensing for GPS-free navigation, and the military interest is not hard to understand. GPS signals can be jammed, GPS satellites can be destroyed, a navigation system based on Earth's magnetic field cannot be turned off by an adversary. If you could build a sensor that matches what a robin's eye can do, you would have a compass that works everywhere on the planet, underground, underwater, in space, without any satellite infrastructure at all. The Navi Sense Cluster of Excellence at Oldenburg, led by Moritzen, just received 54.7 million euros in funding over 7 years, bringing together around 80 researchers across biology, chemistry, physics, computer science, and even social sciences. The project launches this year, and its mandate is explicitly aimed at translating biological quantum mechanisms into technological applications. The project's own description says it plainly. If we can understand how these quantum mechanisms work in the wet, warm, noisy environment of a cell in a bird's eye, this should have huge technological impact. 54.7 million euros for studying how a songbird's eye works. That is how seriously the scientific establishment now takes this, and that gets at the deepest question raised by this entire story. How is any of this possible? Quantum computers need to be cooled to nearly absolute zero to maintain quantum coherence for about 100 microseconds. Yet the radical pair mechanism in a bird's eye requires spin coherence lasting 1 to 100 microseconds in an aqueous environment at 37 degrees Celsius, surrounded by the constant chaos of a living cell. For decades, physicists said this could not happen. The thermal noise would destroy any quantum state long before it could carry useful information. The answer appears to be that biology does not protect quantum states through isolation, the way engineers do with supercooled circuits in vacuum chambers. It protects them through structure, the Cryptochrome protein's hydrogen bonding patterns, its amino acid arrangement, the precise geometry of the tryptophan chain. All of these create a microenvironment where decoherence is slowed just enough for the mechanism to work. The protein is not fighting physics, it is using physics. The thermal noise that engineers try to eliminate is actually, in some models, helping the quantum process. This concept, called environment-assisted quantum transport, is one of the most exciting ideas in theoretical physics. Right now, the noise does not destroy the quantum state. It nudges it in the right direction. Eric Gauger at Heriot-Watt University has noted that behavioral evidence from the radio frequency experiments implies coherence times of at least 100 microseconds. Incredibly long for quantum systems, he says, especially in biological conditions. To put this in perspective, the most advanced quantum computers in the world achieve coherence times of a few hundred microseconds, and they operate at 15 millikelvin, colder than deep space. The bird is doing something comparable at 37 degrees Celsius while flapping its wings. This is not entirely unique in biology, but it is by far the most dramatic example. Quantum coherent energy transfer in photosynthesis. The famous Engel and Fleming experiments at Berkeley in 2007 initially seemed to show that plants were using quantum effects to optimize solar energy capture, guiding photons through molecular complexes with efficiency that classical physics could not explain. The discovery made headlines and launched a wave of enthusiasm for quantum biology. But subsequent work revealed the coherences were likely vibrational rather than electronic, meaning they might not represent the kind of non-trivial quantum effect that the initial hype suggested. The photosynthetic coherences also lasted only about 660 femtoseconds. That is less than a trillionth of a second. Impressive in physics terms, but vanishingly brief in biological terms. Quantum tunneling in enzyme reactions is better established. Enzymes like alcohol dehydrogenase appear to transfer hydrogen atoms through energy barriers faster than classical transition state theory predicts, with the particles essentially passing through the barrier rather than going over it. But these tunneling events operate on femtosecond to picosecond timescales, billionths of a millionth of a second. And there is Luca Turin's vibration theory of olfaction from 1996, which proposes that we smell molecules by detecting their quantum vibrational frequencies through inelastic electron tunneling, though the evidence for this remains decidedly mixed. The radical pair mechanism requires microseconds, 1 to 100 microseconds of quantum coherence. That is orders of magnitude longer than anything else in quantum biology. If it is confirmed, it is not just a quantum effect in a living system. It is the most impressive quantum effect in any living system we have ever found, by a wide margin. A 2025 analysis by Kominis and G. Kaudinakis calculated that biological magnetoreceptors operate near the quantum energy resolution limit bounded by Planck's constant itself. Evolution, working blind over millions of years, pushed these sensors to the edge of what the fundamental laws of physics allow. A 16-gram bird flying through darkness over an ocean carries in its eyes a device that operates at the theoretical maximum performance allowed by quantum mechanics. I want to sit with that for a second, because this is not just a story about birds. In 1944, Erwin Schrödinger, the same physicist who gave us the thought experiment about the cat in the box, published a short book called What is Life? In it, he asked whether the molecular machinery of living organisms might obey quantum rules that we do not yet understand. The question haunted biology for decades. It helped inspire Watson and Crick's discovery of DNA. It launched entire fields of biophysics. But most biologists eventually concluded that quantum effects, the spooky, fragile, probabilistic stuff that governs atoms and subatomic particles, were irrelevant to life as we actually observe it. Too fragile, too quickly destroyed by the warm, wet chaos of cells. Life was chemistry. Good, classical chemistry. The quantum world stayed in physics departments, safely refrigerated. Jim Al-Khalili and John Joe McFadden, in their 2014 book Life on the Edge, articulated the key distinction. All of chemistry is at bottom quantum mechanical. Every covalent bond, every enzymatic reaction, every protein fold involves quantum effects. But that is what they call trivial quantum biology. The water in your coffee mug is governed by quantum mechanics too, but nobody calls it a quantum beverage. What matters is whether living systems exploit non-trivial quantum effects, things like coherent superposition, quantum tunneling through classically forbidden barriers, or spin dynamics where the outcome depends on maintaining quantum coherence for biologically meaningful timescales. The radical pair mechanism, if confirmed, is exactly that. Not just quantum chemistry happening to occur in a living cell, but quantum coherence being actively maintained and exploited by a protein that evolution shaped for this purpose. The birds suggest otherwise, and they are not alone. Monarch butterflies use a cryptochrome-dependent inclination compass for their annual 3,000-mile migration from Canada to central Mexico. And a 2021 study in Nature Communications confirmed their compass relies on a different version of the protein, Cryptochrome 1. Sea turtles appear to use both a radical pair compass and a separate magnetite-based map sense. A 2025 study showed that magnetic pulses disrupted the turtles' map, their ability to estimate position, but not their compass, their ability to choose a direction. Two different magnetic senses, two different mechanisms working in parallel. The mechanism shows up across migratory songbirds, from garden warblers to Australian silvereyes to Eurasian blackcaps. Over 20 bird species have been confirmed so far, and the list keeps growing. Interestingly, species that do not rely on the radical pair mechanism seem to use fundamentally different physics. Spiny lobsters have a polarity compass. They detect which way is north and which is south, which is consistent with magnetite crystals rather than radical pairs. Zambian mole rats are unaffected by radio frequency fields at the Lama frequency, confirming their magnetic sense runs on a different mechanism entirely. The emerging pattern is that radical pair magnetoreception is primarily a vertebrate and insect phenomenon linked to cryptochrome and light, while magnetite-based systems serve different or complementary functions. Many species likely use multiple systems simultaneously, the same way they use sun, star, and magnetic compasses all at once, cross-referencing different sources of information to build a navigational picture more reliable than any single compass could provide. If cryptochrome-based magnetoreception is real, and the evidence at this point strongly suggests it is, then evolution did something that engineers are still struggling to do. It built a quantum sensor that works at room temperature, runs on sunlight, weighs practically nothing, and has been field tested across millions of years of migration routes spanning entire hemispheres. The sensor does not need calibration. It does not need maintenance. It repairs itself because it is alive. It passes the blueprints on to the next generation through DNA, updating and optimizing the design through natural selection with every breeding cycle. And it does all of this by exploiting a quantum mechanical effect that the smartest physicists of the 20th century believed could not survive outside a vacuum chamber. That is not a metaphor. That is not poetic license. That is, as far as we can tell, what is actually happening every autumn when a European robin weighing 16 grams takes flight in Scandinavia and heads south through the night, navigating by a quantum compass in its eyes, following the angle of invisible magnetic field lines that thread through the atmosphere and down into the Earth, guided by electron spins oscillating between singlet and triplet states in a protein called Cryptochrome 4, in a retina that functions as a room temperature quantum computer, more sophisticated than anything humans have built. The story is still being written. The signal transduction pathway from radical pair chemistry to nerve signal remains unknown. No one has directly observed the mechanism working in a living bird's eye, alternative explanations cannot be ruled out entirely. 5 major unsolved problems remain, and the skeptics are doing their jobs, which is how science is supposed to work. But the trajectory is clear. From Schulten's rejected paper in 1978 to the €54.7 million Navisense initiative launching this year, from a dismissed idea about chemistry to engineered quantum sensors working in living cells, the quantum bird compass has gone from wastepaper basket to the cutting edge of physics, biology, and technology all at once. A 2025 analysis calculated that biological magnetoreceptors operate near the quantum energy resolution limit, bounded by Planck's constant itself. Evolution, working blind over millions of years with nothing but random mutation and natural selection, pushed these sensors to the edge of what the fundamental laws of physics allow. That is not a metaphor either. That is a mathematical result published in a physics journal. And the next time you see a robin in your garden, I want you to think about what might be happening inside its eyes. Because somewhere in that tiny skull, in a thin layer of cells at the back of the eye, photons are striking proteins, electrons are hopping along chains of amino acids, radical pairs are oscillating between quantum states at megahertz frequencies, and Earth's magnetic field, 50 microtesla, a force so weak you will never feel it, is being read like a map by a machine that evolution built out of nothing but carbon, nitrogen, oxygen, and 400 million years of trial and error. Scientists used to believe that quantum effects could not survive in biology. They used to believe that the warm chaos of a living cell would destroy any quantum state before it could do anything useful. They used to believe that the future of quantum technology lay exclusively in billion-dollar laboratories with supercooled circuits and perfect vacuums. A 16-gram bird flying through the dark over an ocean it has never crossed before, guided by the angle of invisible magnetic field lines detected through quantum spin dynamics in a protein in its right eye, suggests otherwise. Birds navigate using quantum mechanics in their eyes. This is not a metaphor. And honestly, if science had just published Klaus Schulten's paper in 1978, we would have known about it a lot sooner. Anyway, that is that. If you want to see how another impossible-sounding physics idea turned out to be real, that video is on screen now. And if you made it to the end of this one, subscribe. I will see you in the next one.